Okay, I guess I can already start with the introduction. So, welcome everyone who's already here to this seminar of Paleoperks. Today we are joined by Jernej Jevšenak from the Slovenian Forestry Institute in Slovenia, who will be talking about climate-sensitive periods of tree growth that are advancing in cold and delaying in dry climates. So the format of today's seminar, especially if you're new here, we will first do the welcome and announcements, what I'm doing right now, followed by Jenne's talk for around 30 minutes. We'll then have a moderated Q&A of around 10 minutes, so you can send your questions to either Ahana, the questions host, or directly in the chat. And then Jenne has agreed to stay for tea time, so if you want to have a more in-depth or a more informal chat with him, feel free to stay after the talk. And then a bit of housekeeping before we start. So Paleoperks values the participation of all folks interested in the paleosciences. And also please remember to abide by our code of conduct during today's seminar. Also, please mute yourself for the duration of the talk out of respect for our speaker. As I said, you can ask questions by chatting to the questions at Paleoperks host, which today is Ahana. You can also post them directly in the chat. And if you want to ask them verbally, you can use the raise hand function and we will unmute you. If you have any technical problems, you can also ask Ahana. We have closed captions built into Zoom if you want to use them, so you can use the CC button to either turn them on or off. And then finally, Paleoperks only runs because we have so many early career researchers that are willing to give a talk here, and we're always looking for more. So if you know someone who could give a good talk, or if you want to give a talk yourself, you can also nominate yourself. Please go to this link and fill out the form. This link will be dropped in the chat. And then we also would really appreciate it if you can fill out a weekly feedback form. This is anonymous, optional, but very encouraged. It's just for demographic information, so we know who is watching these seminars. This will also be dropped in the chat. Then a bit more about the speaker today, Jernej Jevšenak. Jernej did his bachelor's and master's in forestry at University of Ljubljana in Slovenia, followed by a PhD in biosciences, also at the University of Ljubljana. He then went on to secure a Alexander von Humboldt postdoctoral fellowship at the Technical University in Munich in Germany, and he is currently a research associate at the Slovenian Forestry Institute in Slovenia. And with that, Jenny, I would like to invite you to share your screen and start your talk.
Thank you, Nina. Okay. So I assume you now see the full screen, right?
Yes, this is a good view. Thank you.
Thank you. Okay. So hello everyone, also from my side. So as Nina said, I'm Jernej Jevšenak from Slovenian Forestry Institute, and Nina already gave some basic information about me. So I would maybe only add that in addition to, let's say, interest in paleoscience, or more specifically in tree rings, I also have worked a lot in other forestry fields, mainly with National Forest Inventory and also remote sensing. But I would say, really the core research interests are related to tree rings. And today I will talk about, let's say, a method which I proposed during my PhD, and now in recent years the method is actually let's say getting developed, and there are new applications which can be used, and I hope that you will get some interesting insights and ideas for your research in the future, right? So basically I will talk about so-called climate sensitive periods, and throughout this talk I will try to develop this idea more and more. But already at the beginning, and I will do this more often, I would like to define this, because climate sensitive periods is not something that's really established term, but try to remember that these are the periods in calendar year, so the season, let's say, where trees most strongly respond to climate. I will end here for now, but later I will try to develop this idea more and more. So I have divided my talk into two parts, and in the first part I would like to talk more about daily and monthly climate growth correlations. And then in the second part, we will get back to these critical climate sensitive periods, and especially what we would like to understand today, it's how they change in time, right? Because we know that in recent decades, a lot of research has been focused on how the climate strength, climate growth correlations are changing in time. But I would also like to show you today that not only the strength, but also the time when trees actually respond to climate is shifting, right? Okay, so there is this discussion about daily and monthly climate. So which climate should you use when you do the tree analysis, dendroclimatological analysis? And I have to say that I'm a big fan of daily data. I say this often, and often we discuss with my colleagues why daily data is maybe more suitable. Sometimes people say less suitable, but today I will try to convince you and show you the application where the use of daily climate data is really beneficial when compared to the monthly. But daily climate data is not something that would be my idea or something that came out recently. So already 25 years ago, there were studies using daily data. So this is a study by Irina Panyushkina. And yeah, already at that time, they realized that if you really want to understand the climate signal, especially the timing of climate signal in readings, then daily data can give you more high resolution signal, right? And then in 2013, so there was this paper presented by Wolfgang Beck, so that Klimtrick, so this is a software, and this was the first software where researchers and students and everyone who was interested in analyzing climate signal in readings, so they were able to use the software to get the idea about when the trees most strongly respond to climate. And then after this paper, I would say that the use of climate data really become popular. And then during my PhD, so this was in 2018, it's now already 10 years ago, slightly less, but still, I published this R package, so the DendroTools R package. This was actually quite spontaneous idea. From a simple function, suddenly, we developed R package, and the package is still regularly updated. There are still new ideas, and we make regular workshops. Usually, this is once a year. In about a month, we have a workshop in Rome in the scope of TRACE conference. Sometimes we also organize independent workshops. So these are usually half a day workshops. But everyone who is really interested in using and testing the DendroTools, I provide here a QR code, and here we have you can see 2-hour workshop from start to beginning, how to use this package, and also the data, the test data is included. So you are invited to test it yourself. Okay, but let's go back to the comparison between daily and monthly correlations, daily and monthly approach when analyzing readings. And if you are using R and readings, then this is something that it's probably very familiar to you. On one hand, we have our chronology here on the left. Of course, this has to be standardized, detrended, and of course, before cross-dated chronology. And very basic question before any dendroclimatological analysis would be which combination of these months, so here we have 12 different months, would be response, the strong, is the most strongly related to our chronology. And usually people start correlating with individual months. So we have here January, February, March, and so on. And then in the next step, one can start to aggregating these months into 2-month period, 3-month periods, and so on. And this is an example from Slovenian sub-Mediterranean side. We often then, especially the dry sites, we often then say that, yeah, the strongest climate signal is related to, in this case, June, July, August precipitation, so summer precipitation. And then we would aggregate and we get correlation coefficient, right? If I illustrate this slightly differently, this is how usually we show climate signal in the Dendrotools R package. It's a heat map, right? So it's a two-dimensional plot, and on the x-axis we have months in this case, and then of course the color represents the correlation coefficient. And we can see, for example, that when we consider individual months, we have very strong signal related to July. But when we then combine months into 2-month periods, then we have a very strong signal in June, July. And of course, we can then aggregate these months into 4-month, 5-month, and 6-month signal. But just I would like you to keep this idea in mind, that the heat map is how we present the climate signal. And on the x-axis, we have the position, and on the y-axis, we have the aggregate, the length of the aggregation window. In this case, we have it in, let's say, on a monthly scale. But in the next couple of slides, I will also show you how we can do this with the daily scale. So this is the daily data, of course, only a part of it. But when we use daily data, how we go with the correlation analysis is basically the same. So if I just show you what is the difference. So let's start here with 92 days, June, August season, right? But when you have daily data, you can easily shift these aggregation windows by 1 day, right? And then in each of this step, we can calculate climate growth correlations, and then we can, of course, plot them using heat maps. And as you can see here, we can then calculate all these possible combinations. And we can also change the size of these windows. We can have narrow windows, and also we can have a wider window. So when we consider all these different combinations, there are a few thousand of different correlation coefficients. And this is how we plot them, right? So if you now have a look at the upper plot, so these are the daily climate growth correlations. And if we look down, so the lower plot, these are the monthly correlations, which we have seen a few minutes ago. And so I hope that you agree with me that the information here, it's very similar. So we have the strongest climate growth effects around summer. Maybe if we even filter out the insignificant correlations, we would then end up with only significant correlations, right? And I think so. The two approaches give us fairly similar information, but when we use daily, we get actually much higher resolution, and we can, as you will see in the next slide, really fine-tune which time of the year this climate growth effects are the strongest. I would also like to point here to this. I don't know if you see my mouse. I assume yes, but here we have with the daily approach, we have around the day of the year 100. We have this significant correlations which are not visible when we use monthly, right? And this is maybe already now an argument, which I will repeat later, that when we use daily approach, we often can also find these subtle climate growth effects, which are narrow, or just because they are they start and end in the middle of the month. For example, April 15, May 15 in this case, approximately, or March 15, April 15. These effects are not visible or not detectable using the monthly approach. Okay, so in the next step, what can we do? So this is also directly a print screen from the DendroTools package. We can first ask, okay, what is the strongest correlation coefficient when we use the two different approaches? And when we use the monthly approach, we see that the correlation is 0.58. When we use the daily approach, we can see that the correlation is 0.65. And I would agree with those who are a bit skeptical. And we can say, okay, these are, of course, daily will always be higher. But what's really important in my case here, that we can fine-tune this optimal time window or critical climate sensitive period. And we can see here that for the daily approach, the time on average when trees most strongly respond to climate is May 27, July 21st, with a length of 56 days. While if we do the analysis using monthly data, we get June-July signal in this case. And in the second part, I think it's again time to remember our critical climate sensitive periods. So in the second part, we will have a look, and we want to study how this optimal time window or critical climate sensitive period change in time, right? Whether they are stable or whether they move, I don't know, towards the beginning of the year, towards the end of the year. So keep this idea in mind for now. I will just give you now, I will show you some results from a study which was published already a few years ago, but I think it's still very important for this specific work, where we used so-called extended European Tree Network from International Tree Database. We have here roughly 1,860 chronologies, mostly conifers, different proxies. And the idea here was that we run monthly and daily response from the DendroTools, our package, for all these chronologies. And then we would simply want to know what is the difference in relative terms between the climate growth effects. We use the EOPS dataset. So this is a gridded climate dataset for Europe and some regions also outside the Europe. And I think it's a really cool dataset. It's observation based, and in my experience, very reliable, especially in the parts of Europe with many meteorological stations, but also for the other parts, with more scarce network of meteorological stations. I think it's very reliable. Okay, so what we did in this case is we have extracted the most significant correlation coefficient for each of these 1,860 training chronologies. And then we have also calculated a simple delta core. So this is the difference between the correlation, the most strongest correlation between the using the daily data and also the monthly data. So, and we compared only windows which have at least of 1 week of overlap, because sometimes it can happen that with monthly correlation we would get summer signal, but when you use daily, you would get early season signal, and this, of course, it doesn't make sense to compare. But in general, as you have seen in previous slides, the identified signals are fairly similar, but they just differ in the strength. And here we have also then extracted these critical time at windows, the onset, end, and length, because we also wanted to see what is the difference on average across this network about these windows that most strongly respond to climate. And we had then also studied distributions of them. Well, there is the main results from this paper. We calculated that on average, the temperature signal is 6% higher. So the correlation strength is 6% higher for daily in comparison to monthly. And for precipitation and spay, it's slightly more, so it's around 7.5. And then we have also investigated why the temperature effects are slightly lower. And the reason is quite simple, because temperatures have very pronounced seasonal cycle throughout the year, while precipitation is much more stochastic, right? So although we have dry and wet periods, but in general, in comparison to temperature, precipitation is still more stochastic, and that's why it matters most when we really find these optimal time windows with precise daily resolution. But yeah, so in general, when you think about monthly and daily, you can expect on average approximately 6 to 7% of higher climate growth effects using the daily approach. And I also promised you and mentioned that we have compared these critical climate sensitive periods using daily and monthly approach. And as you can see here, so we have the onset. So this is the day of the year, the onset day of the year, the end day of the year, and then also we have the length. And it was actually quite surprising to see that the onset and end, they have very similar distribution. We see some minor differences, but in general, these are similar distributions. But when we look at the length, we can see that when we use daily approach, in many cases, we can identify very significant short climate growth effects, which are not detectable or somehow mimicked when we use a monthly approach. And yeah, so this is, would say, an important, especially when you work in northern latitudes or in very high elevation sites where the growing season is shorter. Of course, I'm not saying here that the growing season and the climate sensitive periods are actually the same, not at all. But of course, if the growing season is shorter, also the period during which trees strongly respond to climate are also shorter. And yeah, so you keep in mind then, especially if you work in high elevation or in northern sites, the daily approach could be very beneficial. Okay, so I'm now going to part two. So, and part two, before we will now develop more the idea of how to study these critical climate sensitive periods in time. But before I go there, I would like to also acknowledge the co-authors of this study, which is now in the review. So, and yeah, it's Flori Babst and Alan Buras, and also other co-authors who provided ideas and insights and helped a lot with developing this. Yeah. Okay, so if we now take a look at one example, I think it's the same one as we had before, but when we use the daily approach, we can first fine-tune and narrow down this pixel, which corresponds to the highest correlation coefficient in this case. Example, May 26, July 20, okay. And in the next step, what we do with this analysis is to simply subset our data into, let's say, shorter time windows. In our case, these are 31-year subperiods, and we repeat the analysis and extraction of these windows for each of these subset. Okay, so critical climate sensitive periods have three parameters. It's the start, it could also be the onset or the end, and then we also have the length, right? And what we want to do, we want to study how these three parameters change in time. Yeah. So in the last, as I said, 10, 20 years, a lot of studies focused on how the climate signal strength changes over time. And it's generally known that, especially in the northern hemisphere, we can see decreasing temperature effects in cold climates, while in generally everywhere we can see increasing moisture effects because of the increasing dryness and atmospheric vapor deficit. So these are, I would say, generally known phenomena, but we don't know how these windows, which most strongly respond to climate change in time. So this is this is the main idea and the main focus of this research. And just to illustrate the specific task for one site, so of course, as I said, we divide our chronology and our climate data into sub-periods. For example, we start with 1950, 1980. We extract the the strongest correlation coefficient, and then we also extract the CSP, the start, the length, and the end. And then, of course, we move our data for 1 year. We repeat the analysis, and then we get, I would say, a very simple table. And what we need to do is we need to analyze temporal trends in the start, length, and end. And of course, for one side, we can use relatively simple linear regression models. But because we used the International Turing Data Bank network, we then, of course, applied linear mixed models with appropriate covariance structure and to account for autocorrelation. But what's really important here is this beta coefficient. So here we have beta coefficient in red square. And this beta coefficient is what we need to understand how these three parameters change in time. And for example, if we get a negative beta coefficient for the start, this would indicate advancement, so that this window is moving towards the beginning of the year. If it's positive, it would mean delay. So the critical climate sensitive period starts later in the year. When we talk about the end, it's of course the same. Negative would be advancement towards the beginning of the year, and delay in the positive would indicate delay. And when we focus on the length, so when it's positive, of course, we can talk about the prolongation, and when it's negative, we would talk about the shrinkage. So, okay. And then when we combine all these three parameters together, we get I would say, very good idea about how these windows are changing in time. Okay, I have already explained this. So we have positive and negative beta coefficients, and positive would indicates so the timing, delayed start, delayed end, or extended windows, while negative beta indicates earlier start, earlier end, and shorter windows. So this is the repetition. And now when we think about different combinations of what are the potential changes, so these are 8 potential changes, but I would like to just highlight a bit this idea so you understand even better, right? So this is the baseline CSP. Let's say that this is the baseline in the, I would say, maybe in the first window, 1950, 1980. But when then we apply our analysis, and when we calculate betas, we can see that these windows might be related to earlier start, delayed end, which would also result in prolongation. This is one example, and then it could also be the opposite. So we can have delayed start, earlier end, and these two would then also result in shorter time windows. But windows might not change in the length, they might only shift left or right. So this is an example for earlier start and earlier end without any change in duration. And then we can also have delayed start, delayed end, no change in duration, or this could be no change in start, but only delayed end and extended duration. So there are also some other combinations, but I just wanted to give you the idea and illustrate how these potential shifts could happen. About the data, I don't want to spend too much time on this because, yeah, it's International Tearing Data Bank. This is a recent study, so we have much more data. And at that point, I would like to really thank all the data contributors who upload data online and actually make such studies possible. We have divided our data into climate zones. So these are Copenhagen climate zones. And we also elaborated on precipitation patterns. So we have dry winter, dry summer, and no dry season. Altogether, we wanted to keep only climate zones where we have at least 100 sites. So we are missing here tropical climate zone and also polar climate zone. We did rigorous, filtering, so quality checks related to the signal strength and also the time coverage and some other typical filters which are used in such large scale studies. We have linked each chronology with the daily ERA 5 length, temperature and precipitation data. And then we have run the DendroTools analysis, as I showed in the previous slides, for each of these drivers, for each of the site across the climate zones. And as you will see, we have also developed models for different clades, so for angiosperms and gymnosperms. And one important is also that we have generally so we have classified all chronologies into first code limited sites. So these are sites where the most significant temperature signal was positive, right? So positive temperature correlations, which indicate that generally these are cold climates, high elevation northern latitudes, and we did specific models for cold limited sites. Then we have heat limited sites. So these are trees that generally respond negatively to temperature because of the heat stress. So this is very common in, I would say, temperate lowland forest. And then we have similar group, but not the same similar. These are drought limited sites, which generally respond positively to precipitation. And these are also, more in dry regions. But as you will see, and as we know, drought limitation is now also very common in also colder sites. So just that you understand that when I will show you the coefficients, that we have separate models and separate coefficients for these three categories, and also for different climate zones and also for different clades. Okay, so we are now already at the results section. Here we have just to guide you through this results. So we see here three. So these are results for code limitation, and we can see that for the CSP start, so we see strongly negative beta coefficients, which indicate advancement for the beginning. Yeah, and we can see also very strong negative coefficients for the end, which also hint that these windows end earlier. And I have here illustrated the shifts which happen in the cold regions, and so we see that the trees in the cold regions now tend to respond to climate a few days earlier, and they also end earlier as in the past, right? So we can only compare the beginning of the analysis, which is 1950, 1980, but these results are related to the cold climates were the most consistent, and whatever model we used depends on the time coverage. We always got these very obvious results, which are also strongly aligned with phenological shift, right? We also see that leaf phenology also shifts towards the beginning of the year. And here we see this go hands in hand. So CSP and also changes in leaf phenology. And Yeah, we have calculated roughly, so this is of course only approximation with some error, that in cold limited regions on average start is shifted by around 4 to 5 days per decade, right? So in 3 decades, 4 decades, this would be roughly 3 weeks of shift in this period, which was used in our study. So this is 1950 2010. Okay, and now we go to the heat-limited sites. So if you recall, heat-limited sites are trees or sites which are generally limited by heat, which means that, yeah, when it's hot, let's say summer, they generally grow less. And what we see here, that here we get the opposite result. So heat-limited sites do not start and end earlier, but they actually start and end later. Yeah, so these results and coefficients are lower. You can see that here we talk about 1 to 2 days per decade, while for the cold limited sites, the shifts were about 4 to 5 days per decade. So let's say three times higher in relative terms. And of course, we discussed a lot, but I think it's pretty obvious that because of the hot summers, because of heat stress during the summer, trees simply have to behave more conservatively, and they wait for better conditions, which is also we assume more suitable for photosynthesis, and then they complete the ring formation, or later in the growing season. So this is our best explanation so far, which is also supported with quite a lot of literature published in the recent years. And we got similar results for so-called drought limited sites. So these are trees that are generally water limited. And here we did not observe very systematic change for the beginning, but what we see are systematic changes for the length and end. So also trees that are drought limited, similar to heat limited, they tend to respond to climate longer in the season, and also they end later. So here we have also coefficients about 1 day per decade. And we also got, as you will see later, quite strong variability among different climate zones. But on average, we still see that there is strong evidence that this response actually ends later in the year, and also lasts longer. And this is also strongly supported by so-called bimodial growth, which is typical for dry climates, Mediterranean climates, where trees during the peak summer heat or peak summer drought, they tend to reduce growth or even stop it, and they continue later when water is available, or when conditions are more suitable. Okay, so I want to show you this. Let's say these are now the same results, but for different climate zones. And I would like to show you that, for example, for the cold limited, we see very consistent results for the start and for the end across most climate zones. When we go to drought limited, we see more variability, but for example, for prolongation, so the extension of these windows, these are, I would say, quite consistent. We see some examples here for the temperate, no dry season. So this would be Western Europe, where these changes are not so significant, but in general, across most climate zones, we see these significant patterns. When we go to heat limited sites, we see even higher variability, but on average, for example, for the end, we see very consistent shifts toward the let's say delay. But as always, there are some exceptions, and especially here, for example, for cold no dry season and cold dry winter, we don't see these extensions for the end, right? So there are always some exceptions, but these patterns which we report here I would say are quite common, but there are always exceptions. Okay, maybe so I'm now getting close to the end. One of the questions which we also asked are differences between gymnosperms and angiosperms. So here I plot changes in CSP across different climate zones, but separate coefficients or separate changes for gymnosperms and angiosperms. And what we can see here, the gymnosperms and angiosperms behave very similarly in different climate zones. If we calculate distances between these 2 points, they will always be the shortest for the clades inside the same climate zone, right? And this is something that indicates that changes which are happening for the vegetation are fairly similar for gymnosperms and angiosperms, but they are not the same, right? Because here we still see differences in terms of the position of these symbols. Um, but still would say that changes inside the climate zone are more similar than changes across climate zones. Okay, of course. So now we could talk about the consequences and about the effects of these changes in CSPs. But I will, because I see I'm already a bit over time, I will just say that keep in mind that when people do climate reconstructions, that in the past, or when we reconstruct, for example, summer temperature, and if we go long enough into the past, we might not always reconstruct this signal for the same period as we do in the present, right? It could happen that, for example, trees a few 100 years ago were not related to June, July, August, but maybe mid-May to mid-August. And of course, this is now something that we have to discuss what this means. But I think that our study nicely shows that the period during which trees respond to climate is not so stable in time as we thought. Okay, for take-home messages, I will just quickly repeat. So we have that code-limited sites generally show shifts toward the shifts of these climate signals towards the beginning of the year. And then when we focus on drought-limited sites, these shifts are slightly delayed for a few days per decade. And this is especially obvious more for drought-limited sites, where we also see prolongation in these windows. Wet sites, I did not really talk about these results, but we see that when we have more wetter sites, although we don't have tropical zones, but we have some sites which are relatively wet, these changes are usually the least significant. Yeah, so we cannot prove that, but generally what we see is that the drier the site, the most stronger are changes in CSP. And as I said, so changes and patterns were broadly consistent across gymnosperms and angiosperms, although we also see differences between them. And so the clade information explains more than the climate information. Okay, and I will stop here now, and thank you for your attention. I hope that you got some interesting insights for your work and for your study. And if you have any questions, I'm happy to answer.
Thank you very much, Jernej. That was a super interesting talk. Reminder to our audience, you can ask questions. If you want to stay anonymous, you can send them to the questions at Paleoperks host. You can also just post them in the chat or use the raise hand function if you want to talk to Jernej. I will just start with one that I personally had. I will also drop it in the chat. So you showed data going back to 1950. So how far back in time can you technically go with daily growth data?
Yeah, yeah. So I would say that most data sets, yeah. So in my case, this was the 9050, right? And Era 5 global data sets start in 9050. Also, EOPS generally starts in 9050, but there is also let's say experimental data set from the EOPS. So this is for the Europe only, which starts in 1920. And of course, we also used it and tested it. But I would say for some of the studies, what we see that if we do the analysis and we go further back in time than 1950, we often get weird results. I would say unexpected. And we see some systematic changes when we go back in time. But suddenly, if you go too much back in time, you would get the results which I would personally not trust. So I would say that for temperature, there are some meteorological stations in Europe, and I'm not familiar so much with other continents, but um, big cities usually they have daily data which goes back in time for at least 100 years. For example, I know for Ljubljana in Slovenia, we have daily data with only a few missing data during the war periods, starting on January 1900, right? So this is I would say quite a lot of data and also very reliable, especially when we talk about temperature data, because temperature data has pronounced spatial pattern, and usually if it's warm in East Slovenia, it will also be warm in West Slovenia or in Hungary and neighboring countries, but precipitation is much more stochastic, and usually we see that precipitation is more difficult to interpolate. So I would say if you do some global analysis, you should probably end in 1950. If you do some local analysis and if you have reliable daily climate data, you can definitely go much further back in time.
Okay, thank you very much. Then Andy Johnson has a question, but feel free to ask.
Thanks. I finally found the raise hand function. That was really fascinating. I'm a sclerochronologist, so I work on, in my particular case, mollusk shells. I'm just wondering what the applications might be in them. But I'd like to go back to the slide that you looked at the consequences. You sketched over it a little bit because you have that's it. I just want to see. What I wanted to ask, and I guess it may be in there, but perhaps you could expand on it. What are there effects on or do you sense that there might be effects on the longevity of trees?
Longevity. Yeah, that's a good question. My my personal best guess, I would say now they are none. Because what I would personally like to, I thought you will ask about productivity.
Yeah,
But generally, I think that these changes are mainly driven by temperature, and this only causes that trees do pretty much the same processes, but in slightly different time. So I wouldn't say that there are any consequences, or maybe you have a better idea, but I would say that these changes only indicate that the ecophysiological processes which are related to transpiration, to wood formation are similar, but they actually happen in slightly different time of the year. So for the cold sites, we see they happen slightly earlier because
yeah,
of the warmth, but I wouldn't, because we also don't really see systematic changes in ring widths in last in ring width, so in last 40-50 years. So like, because many people would expect that because of longer growing seasons and because of warmer temperatures, the trees will now grow more. But in what we see, or what I maybe believe most, is that trees are still very conservative, and they don't keep growing. Even if they could, they behave more conservatively, and they maybe start saving carbohydrates for the next year, because you never know what is going to happen into the next year. So I don't see consequences for longevity, but yeah, this is only my guess.
What I'm kind of edging towards is, can you see potential changes in community structure?
Mm, not sure. Not
Yeah.
sure.
Too
Not
short a
sure.
time range perhaps to tell. That it kind of relates to the question that Nina was asking about, the time scale over which this sort of work might be done, and whether if you could do it over a longer time scale, you might be able to tie
Yeah.
in broader scale from,
Yes. So these changes, in my opinion, are strongly related to phenology changes.
yes.
Because I was discussing with a colleague recently, and he said that they see fairly similar changes when you analyze trends in leaf phenology, and they also see very consistent changes for the cold climates, but when they go into dry climates, things are more blurry. And so I believe that, okay, in my opinion, trees passively react to changes in the environment and they adjust. But yeah, so far I don't see, or I cannot come up with consequences for vegetation dynamic, or
Mm,
yeah. But
mm.
it would be interesting to link this if there are any changes in productivity. So this is something that I would like to do, but also, yeah, we need productivity. It's also a complex question, and um, how to really, yeah, because it's only radial growth, so we can always link these changes to radial growth. But for example, what we also tried to see if there is any systematic relationship between beta coefficients, these changes, and the ring widths, and we didn't see anything. So yeah,
Yeah, so they might be in another dimension, they might be behaving differently. They might be growing, you talk about radial growth there, but there
yeah,
might be variations in upward growth.
yeah. We can,
Yeah, not that you can
yeah.
address that easily. It's,
No, this is really scarce data, because radial
yeah,
growth, and actually it's really hard to measure it. I mean,
yeah, yeah,
even with maybe now with the laser scanning, and the yeah, with the terrestrial lasers and airborne lasers, we can get much better information about the height growth, but still there is high uncertainty when you measure it, because it's a few centimeters.
yeah.
With that,
Okay.
I am going to mute you for now, Andy, but feel free to stick around for tea time, because I also want to ask Ru Huang's question, who posted in the chat. So Ru Huang asks, he has two questions. Thank you much for a nice talk. First one is, how do you consider the influence of climates apart from the periods revealed by the daily climate growth relationships? In my understanding, daily climate tree growth relationships primarily identify the critical climate windows that constrain radial growth rather than the true beginning and end of tree growth itself. And then the second one is, have you considered other climates besides temperature and precipitation?
Yes, the first part I definitely agree. So, and maybe I should highlight this more, but critical climate sensitive period, it's not the same as growing season. I mean, there are different definitions of growing season, but if we say that growing season is the beginning of food formation and the end of food formation, then critical climate sensitive period would normally be somewhere in between. Yeah. So usually this would be summer or early season, which most strongly drives climate signal. And this is not the same as growing season. So that would be my first answer. I think I hope I clarified, but if not, let me know. And the second one, I forgot. So, can you please repeat the second part of the question?
Yes, of course. Have you considered other climates beside temperature and precipitation?
Yes, we did all the analysis also using SPEI and VPD. So for SPEI, so this is the standardized precipitation evapotranspiration index, yeah, based on the water balance, and so it's SPEI behaves exactly the same as precipitation. And also at the beginning of my talk, I showed some results when we compared climate growth effects between daily and monthly, and there we also use SPEI and precipitation and temperature. And SPEI really it's very similar to precipitation signal. Although VPD, when we plot these CSPs into two dimensions, VPD is somehow in between. So we then did not really elaborate highly on changes based on VPD, but we decided to keep only temperatures because, yeah, temperature were very important for these cold climates, and also precipitation, which explained a lot about what is happening in the dry climates. But we could easily also show what is the VPD, but VPD is somehow in between these two drivers, and it's more difficult to explain. So far we did not then decide to include VPD, but if needed, we could also show it.
Okay, and Hu Huang says, thank you so much for your explanation. It's very clear now.
Thank you.
Meanwhile, I received one anonymous question in the chats asking, have you considered doing any dendroecological factors when doing daily climate response analysis?
So dendroecological factors. Yeah, so the thing is that usually when tree people go and collect tree data, so tree cores, usually that's all what we collect, right? So, and only later when we think about the networks, we see, okay, it would be really great to have also soil data or maybe some stand parameters, stand density, precise species composition, but this is really rare. And yeah, only in big European projects they collect more data. But so far, what we more or less then opportunistically, if we see with my colleagues that we have maybe appropriate data, as I'm at the moment, I'm in part of study where we have the same species, but then we have different waterlogging sites, right? And this could be an example that where we then study the waterlogging effects on climate growth correlations, and of course we see differences, but usually we don't really have a lot of data to really explain that. And so my answer would be I would love to, but just would need more data to really do that. And maybe, yeah, because these networks are often based on different groups, different projects where everyone has, different purpose. Everything what is in common are tree rings, which are always comparable, but then we really lack local site data that would allow us to tell us more about the signal. But maybe what I can mention is related. So for example, I'm now supervising a PhD student, and what she does is, so she collected approximately 25 sites across Europe, and for each of these sites she measured precise competition for each tree. And now what we can do is we can study the competition effects for each tree in this network. And this is something that we can already see can add and explain more, not just site level, but we actually see competition level differences in climate signals.
That sounds really cool. I think that's a very fair answer. Right now, I have one more question, and I think this will be the last one because we're at the top of the hour. And then we can move into tea time if there's interest. So this one is asking, regarding the climate zone data set that you showed, Why is tropical data missing? Is it more difficult to sample tropical trees?
Oh, maybe to sample it's not so difficult, but to measure it's definitely much more difficult. So I'm not an expert in tropical tree rings. There is a community that is now actively working in this direction. So this is the Tropical Tree Ring Network. And we have already discussed with them to also run the analysis in the tropical zone. So I'm very very confident and let's say optimistic that hopefully in 2 years, maybe 3 years, usually time is a bit faster than we think, that we can also show the results for tropical network. But there is a science paper, I think it was published last year from Peter Zudema and colleagues. You can definitely see, and they reported that the drought effects are, I think in the title they wrote, not so significant, so that they are relatively minor drought effects so far in the tropics. So I would expect that changes in CSPs are relatively small in tropical zone, but maybe I'm wrong. But yeah, so but it's not so easy to measure the rings in tropics, but it's definitely possible.
Okay, that's good to know. Again, with that, because we're at the top of the hour, I would like to end the Q&A. Thanks a lot again, Jernej, and also
Yeah,
thanks
thank
to the
you.
audience for asking very interesting questions. Before we go to tea time, I will just share a bit more information about next week's talk. Yeah, so thanks again, everyone, for joining us. And thanks, reminder again, we would really appreciate it if you fill out a weekly feedback form. Next week on May the 5th, we have a talk by Chris Harris from the University of Münster in Germany, and he will be talking about ichnology, specifically Devonian ichnology, and notes the time difference again. So this one will be again at UTC 15 o'clock. Then, as I mentioned before, up next is tea time with today's speaker, which is just an informal conversation about the talk in a relaxed setting. So everyone can turn on their cameras if they want. We will stop the recording and you can just continue chatting or ask about a bit more technical questions if you want. Before that, we want to give Jernej the time to relax a bit because he has been talking for quite a long time. So we will take a 2-minute break. And after that, we will be back for tea time. So hopefully see you there.
Okay, thank you.